Good afternoon, everyone. My name is Ogoti. I'm a postdoctoral research fellow at the Center for Epidemiological Modeling and Analysis, found at the University of Nairobi. I'm a trained virologist, that's my core, but over the last years, I've found myself using a lot of computational methods, that's bioinformatics, and a couple of other methods in mathematical modeling. We just switch focus to something else. So talking about Middle East respiratory syndrome coronavirus, a project that formed part of my PhD. Just to give you some context, emerged in 2012 from a patient in Saudi Arabia. To date, we have seen around 2,600 laboratory cases, including over 900 fatalities with around 36% case fatality ratio. And most of those cases reported have been reported in 27 countries, most in the Arabian Peninsula. And this is just a time course chart, just showing the reported cases since up to April 2024. The big spike around here is an outbreak that occurred in South Korea, which was an imported cases from someone traveling from the Arabian Peninsula. And interestingly enough, in the year 2020, the cases kind of tapered down or disappeared, but this is potentially an artifact because most of the responses or the resources were committed to SARS-CoV-2 surveillance. So this is just the epidemiology, and in the African continent, we mostly have the camel or dromedary infection, which is the reservoir host of the virus. In the Arabian Peninsula, we see a spillover from the camels to humans, and part of like South Korea, we saw human-to-human transmission, which was mostly a hospital-acquired infection. So far, what is interesting and what got us interested in this study is we have seen a long-term and widespread circulation of MERS in dromedaries. There is literature or there is evidence that samples collected around 1983 from Sudan and Somalia had neutralizing antibodies to MERS, that these are camel samples. And just for context, the first case was detected in 2012. So far in the African continent, what makes it interesting is that most of the dromedaries, so the camels, around 70% of the total population is in the African continent. So far, we have only recorded 6 human cases with around 0.10 seroprevalence. Most of them, there are no primary disease. In the camels, we have a similar kind of seroprevalence and virus prevalence. It's very comparable between the Arabian Peninsula and Africa. We only see unidirectional camel trade, so most of the camels which end up in the Arabian Peninsula come from Africa, specifically the northern horn of Africa. Studies on T-cell responses have been done just looking at the other marker, and so far it's been done in Nigeria. And also in terms of genomics, we see 2 different viruses. In the Arabian Peninsula, we have clade B, and also in the African continent, we have clade C. So the question is, from this paper done by colleagues, we see these transmission dynamics, and you can see in the blue color, these are the transmission dynamics of the virus between several African continents in blue, and only we have a an orange color where we have the lineage B5, which kind of circulates or dominates in the Arabian Peninsula. So the question is, why are the human outbreaks usually restricted in the Arabian Peninsula? So for this, I had 2 questions, and I will take you through quickly. So the first question was, what are the MERS infection patterns in Kenya, which is probably the epidemiology? And the most important key question, why do we see limited human MERS-CoV cases in the African continent? For this, we set up in the year 2022 and 2023, we set up a project in Kenya, in the northern part of Kenya, where over 1 year, we sat and sampled camels and tested them for MERS RNA and antibodies. So this is just a general map showing the origin of where the most camels originated, sampled over 1 year. That is the northern part of this, just a map of Kenya, and most of the camels originated in the northern part of Kenya, which borders Ethiopia. What are the key findings? One of the interesting findings was over the 12 months that we were there, we got 36 RNA positive camels. And this just represented, and what was interesting to us, these findings were only found in specific months. That is September and October 2022. We saw no cases there, and then other cases rose up in the month of January, February, and March. This kind of concluded to us the incidence, or the biphasic incidence. The virus doesn't seem to be endemic, kind of comes in batches or outbreaks. And we tried to correlate this with rainfall, which was quite useful. And we could see that, and we saw that in the areas where we have less rainfall or the dry season, there is actually high risk of infection, which makes sense because during these times, most of the camels congregate and we have high transmission events. What happens to the workers? So we did this study in abattoir workers, and the abattoir workers are classified as occupational hazard. And within around 48 abattoir workers we sampled, 7 of them had antibodies, and out of 1 we actually checked for neutralization, and he had neutralization antibodies towards MERS. So this is just most of the findings in this paper. You can go look at it. But for me, what was more interesting was, why don't we see actually human cases in the African continent? It's a question ever since MERS came up. It's been a question battling the mask of researchers. So the idea is, so we have in our collaborative group, there's this paper which came out this year looking at the ongoing evolution of MERS in Saudi Arabia. And interestingly enough, so these are all the lineages. We have C, which comes from Africa, and this is lineage B5, which is very dominant in the Arabian Peninsula. And you can see the ladder-like evolution with a very nice constant molecular evolutionary rate. That's the lineage B5 sequences alone. And for the sequences, so we were able to sequence 12 full genomes from the RNA we got. And they clustered into 4 different clusters, which is circled in the red color. Interestingly, the clade C in Africa is quite diverse compared to the B, with a couple of strains. Currently in the Eastern Africa, we have the clade C2 dominating, circulating, and these were most of our sequences clustered. Out of that, we were able to isolate 6 viruses for further phenotypic work, and also we were able to do the molecular clock analysis for our sequences, and we could see a very constant evolutionary rate, similar to the Arabian Peninsula. But interestingly, when these viruses were subjected to growth kinetics in the lab, you could see the African or the Kenyan viruses were replicating at lower titers compared to the Arabian clade B or the original prototypic clade A. So for us, what was interesting was that when we tested these same viruses in the nasoepithelial cells, that is the first contact point of viruses, we could see that the Arabian Peninsula replicated at a high titer compared to the Kenyan viruses, and got us wondering, could it be the reason of the observed phenotype of the African viruses that they cannot actually attach well before they cause infection? And for that, we did what we call protein stability, in silico protein stability analysis, based on a tool called Foldex, which is very popular. It calculates the empirical force field or the protein stability changes based on the free Gibbs energy. And this is just the equation where it takes your wild type versus the variant and calculates the delta of the free Gibbs energy. And based on the mutants that we saw in the Kenyan variants, that is the spike, we were able to see that most of the mutations that we're able to see in our spike, which is of course the first point of interaction, the human receptor, that there were more destabilizing variants. And specifically for us, since I'm from a virological background, I took two of them, this one, which are basically coming from the receptor binding motif, did a site-directed mutagenesis, introduced it in this system called the virus-like particles, and I could see it clearly in the EMC, or the prototypic in the Arabian Peninsula. Once I introduced these mutations from the Kenyan variant, it replicates at a lower titer compared to the parent. This kind of goes to the summary of this talk, that one, from the epidemiological point of view, the incidence might be seasonal. Of course, there are many factors working to that. And then we have this unique clade, that is clade C2, circulating in the last 2 years in Africa, meaning it's kind of the predominant strain. And we know, of course, the clade B has enhanced replicated fitness compared to the African clade C. And generally, from this point, we see that the spike, or the clade C spike, plays a very big role in the phenotype, but generally a combination of factors maybe are responsible for the lack of the human cases in Africa. Just as a bonus point is we are developing a mathematical model just to be able to integrate the mass genomic data by explaining what are the infection pattern of mass in camel herds in East Africa. And this is work in progress and will be seen in the next few months. Thank you very much. I would like to acknowledge my collaborators from Berlin Charité, where I did this work, and SEMA, and my working group in Kenya. Thank you.
Great. Do you have any questions for Brian?
Thank you for a very interesting talk. It was quite easy to follow along, not being from an epidemiological background. So I'm curious, do you think there's a major risk of the bee clade spreading from the Middle East into African camel populations? Or do you think there are some environmental factors that prevent it from taking hold in Africa?
Thank you for the question. I think the reason, or the main reason why you're not seeing the bee clade dominating in Africa is because of the unidirectional trade. So camels go one way, they don't tend to come back. And that's one of the biggest drivers why you don't see the bee. But also there is evidence that some of the environmental factors, and most interestingly, the clade C kind of is the parent which gave up the bee in a way. But I think the lack of unidirectional trade back to Africa is one of the biggest reasons I don't see that. Yeah.